Hello! This week we're going to talk about regulation of gene expression. We're going to talk about this in both prokaryotes as well as eukaryotes. And both of them are going to have a little bit different mechanisms of how they accomplish this goal. So in prokaryotes, remember they don't have a nucleus. So as soon as they have an RNA for a product, they can immediately begin to translate that. They're mostly going to be regulating things at the transcriptional level. So for prokaryotes, they will tend to cluster genes together in this operon model. In this model, they're going to have first a promoter. This is a short DNA sequence where RNA polymerase will attach. They will also have an operator region. This will be a DNA portion where the active repressor can bind. And then you can have structural genes. This can be one, two, several genes that will be coding for enzymes. And typically, different metabolic pathways will be translated together as a unit. And that way, everything that an organism might need for a particular pathway will all be there at the same time. Bacteria adapt to their environment by producing certain enzymes, and we're going to call this inducible. If something can be turned on, it's going to be known as inducible. So they're only going to be turned on, this product's only going to be made when specific substrates are present. If enzymes are continuously being made and it doesn't really matter what else is in the cell or what else is in the environment, we'll call this constitutive enzymes. And so constitutive just means that they're constantly being made, no matter what else is there. Typically though, if you have a lot of an end product, a particular metabolic pathway or different pathways, maybe you have a lot in the environment, this will typically repress gene expression of that particular product. And this is known as a repressible system. Regulation of either of these types, either the inducible where a set of genes is turned on or repressible where a set of genes is turned off, themselves can be under positive control or negative control. If something is negative control, it means that gene expression is going to keep going unless it gets shut off. And it's usually shut off by some form of a regulator molecule or repressor. In positive control, transcription is usually off, and it's only turned on if a regulator molecule directly stimulates that RNA production. So it's only usually turned on when an organism is going to need it. So here's a visual of the operon model. Typically upstream, maybe quite a ways upstream, we'll have a regulator genes. This will code for mRNA, which will code for, in this case, an active repressor. Here's our promoter, here's our operator, here's our structural genes. When this repressor is active, it goes and it binds this operator region. When there is a repressor bound to the operator, there is no transcription that can happen. This transcription is prevented. RNA polymerase may come and may sit down on the promoter, but it can't physically move past this repressor, and so it's going to be blocked from doing any transcription. The Lac operon is a classic operon that's used to describe bacterial gene control. In this particular operon, there are going to be three structural genes. They're all next to each other on the chromosome, and they're all under control of a single promoter and a single operator. There's a regulator gene that codes for a Lac operon repressor. It's normally it's normally going to be bound to the operator and is normally going to prevent transcription. So this is something that it's there, but it's usually not being made. It's usually turned off, this operon. However, when lactose is present, now suddenly this organism needs the genes to digest lactose. So lactose can bind to the repressor. The repressor is going to undergo a conformational change. It no longer can bind to the operator. So we can say that lactose is the inducer of the Lac operon. These enzymes are inducible enzymes. This entire unit is an inducible operon. So in the absence of lactose, the repressor is made. These enzymes are not needed. The expression of the genes encoding these enzymes is thus repressed. If lactose is present, lactose can bind to the repressor, stopping it from binding to that operator, and it can then induce activation of genes. Once all that lactose is metabolized, or we don't need it anymore, there's none left or able to bind to that repressor, and that transcription will cease. So here's a schematic of this. Again, in the absence of lactose, there's an active repressor that's made. It will bind to the operator region. RNA polymerase may come to the promoter, it may bind the promoter, but it can't do any transcription of these downstream genes. In the presence of lactose, it will bind to the repressor. This is going to cause it to have a conformational change, and it can no longer bind to this operator region. Now, RNA polymerase can come to the promoter, it can sit down, it can bind, and it can do transcription. Because transcription is being made, there is no inhibition on the translation, and it can immediately begin to do translation and produce the protein products, the enzymes from this particular structural genes. In the trip operon, it's going to basically be the opposite of the lac operon. In the trip operon, this is going to be an operon that is usually turned on. This is going to be coding for tryptophan. So it's usually on. However, if the cell finds itself with an abundance of tryptophan, it doesn't make sense to make more tryptophan, and instead it will shut off production until the level drops. So in this case, the repressor codes for, or excuse me, the regulator codes for repressor is normally unable to bind to the operator. Okay, so RNA polymerase binds to the promoter, the structural genes are expressed, and we get the product enzymes that are needed to produce tryptophan. If there's plenty of tryptophan, the cell doesn't want to waste energy trying to make more tryptophan, these enzymes aren't needed, and so this operon turns off. In this case, tryptophan is a co-repressor and helping to stop production of itself. So here's what this looks like. Again, in the absence of tryptophan, there is a repressor made, but it's not active. This allows RNA polymerase to come and sit down on the promoter, allows it to do transcription, to make the mRNA, and then translation and produce the protein products from these structural genes. These are then going to be used to create tryptophan. If the levels of tryptophan are very high, there's enough around, it's just floating around the cell, there's plenty of it there, it will bind to this repressor, activating it. Okay, now we have an active repressor bound to this operator region. Now, RNA polymerase cannot sit down, it cannot do transcription, and the levels of tryptophan then can level off because no more tryptophan is being made. Attenuation is an alternative repression mechanism, and this can be thought of as translational regulation of transcription. So I said mostly these prokaryotes, they're regulated at the transcriptional level, but there can be some regulation at the level of translation. So what happens here is this leader region can form two different conformations, and it really just depends on the presence or absence of tryptophan. And in the presence of tryptophan, the hairpin structures formed act as a terminator. So let's take a look at this. Okay, so here's this initial stem loop structure that can be formed. You can see the tryptophan codons here, regions 1, 2, 3, and 4. If this charged tRNA is available, the ribosome is going to move. Okay, you can start this translation, but it will terminate. There's going to be this 2-3 stem loop is prevented from being formed, but you will not be able to form any of these downstream regions. If this charged tRNA is not available, this ribosome is going to stall on these tryptophan codons. This allows it to form a 2-3 loop. At this point, transcription can proceed. And so really it just comes down to, is that tRNA containing that tryptophan, is it available or not available? If so, different things are going to happen there. So some differences between these, because they're similar but yet different. In the trip operon, this is an example of negative control. Again, you don't need to make tryptophan unless these levels are low, and then you need to make more. Why waste it? Okay, if you got tons of tryptophan laying around, you don't need to make more. That just doesn't make sense. The cells try to be as efficient as possible. In the lac operon, this is an example of an inducible operon. In this case, this is a positive control. It's usually off, and you don't make these enzymes to digest lactose unless you need the enzymes to digest lactose. If you do, there's no other energy sources around like glucose, now this operon can be turned on. Now I said I'd talk about the similarities and differences, so these are the differences. In both cases, though, if a repressor is bound to the operator region, you're not going to get transcription happening. It's just at what point are you going to be inhibiting it? Is it usually on and you're turning it off in the case of this? Or is it usually off and you're turning it on in the case of this?